Well, my name is Mark Edlin. I'm going to introduce our Diatom Web Academy today. Sarah will handle introductions of our speaker, and Sylvia will monitor our chat room with people's questions both during the presentation and also after the presentation. So first, welcome to everyone. I hope everyone's friends and families and everyone is doing okay during these crazy times. You're joining us on the Diatom Web Academy. The Diatom Web Academy is sponsored by the Taxonomic Certification Committee of the Society for Freshwater Science and also the diatoms.org website. We want to use this platform, especially right now in these pandemic times, to allow us to connect with our friends and connect with our colleagues in times when we're not holding meetings and not able to do that. We can do that. We can use this platform for that, but also as a mechanism to get out information, in particular information that's relevant to the taxonomic certification process that we're developing in North America for diatoms. All of the webinars that you're seeing here are available on a YouTube channel for later viewing. David Williams will be up in a day or two. And all of those can be found either by going to the diatom.org or diatoms.org website in the news section to find those, or you can go to YouTube and just search on Diatom Web Academy to see all of those for later viewing. During the presentation, please feel free to answer or ask any questions or concerns that you have in the chat room. It'll be monitored by Sylvia, and then after David's presentation, we'll be hosting a discussion and questions afterwards where you can ask more questions as well. With that, let me turn the mic over to Sarah Spalding to introduce today's speaker. Thank you.
Thanks, Mark. And I'm pleased today to welcome Dr. David Williams, who is a researcher on diatoms at the Natural History Museum of London. And I appreciate David coming to us today to talk about his work. He is a specialist on systematics, phylogeny, biogeography, classification. He's done many of the revisions of the group of A-raphid diatoms, including Fragilaria, Cynedra, Diatoma, Meridian, and many others. And so today he is going to talk about diatom classification and how it ties in with identification and phylogeny. So thanks so much, and I'll turn it over to you, David.
Okay, thank you. My mic's on, yes?
Yes, you're on.
Good.
So, great.
Good, good. First thing I should do, I need to in return, thank the organisers for inviting me to do this. I am genuinely flattered to be asked, because I've attended some of these and they're going to be tough acts to follow. So I'm going to do my best to tell you something or other. I guess most of what I tell you, you may probably already know. And I'm going to concentrate just on a few things that interest me, with the view that I might be able to interest you in the things that interest me. I have spent, let me start as a preamble. I'm going to tell you something about classification in general, some experiences of mine. I said that it was going to be some stories. It'd just be one story, but first I should say that I've spent most of my working life at the Natural History Museum. That's obviously been an immensely rewarding time being there, not just because I've been able to interact with some of the great diatomists like Pat Sims and Bob Ross, but the fact that the museum has several departments: entomology, botany, paleontology, and I could interact, I still do interact with a lot of different people who all study different kinds of organisms, but all focus more or less, or used to focus more or less on the subject of classification. And that's immensely useful to be able to talk to people, work out stuff, see how we all do it, see whether we agree with each other, and so on. that has been phenomenally useful for me to have such an interesting bunch of people to interact with. My story relates to a different kind of interaction, an interaction that I don't normally have. I don't have a lot of interaction with the people down in the museum itself on the exhibition side, but this interaction stayed with me. What I tell you is as far as accuracy, is a truthful story. It actually happened to me, and you will have to believe me that I learnt a great deal from this interaction, and it stayed with me. I don't know how many years ago this interaction happened, but this actually stayed with me. Let me do something here. My slides aren't going. Shit. Sorry, I don't seem to get my slides moving.
Did you try? Oh, there you go.
Oh, I got it. All right. There was, at the time, it's gone now, there was a British natural history exhibition in the museum. This image is not it. This image is our bird gallery. I used to have to walk through the British Natural History Gallery every morning on the way to the cafe because it was the only way to buy coffee in the museum. This was the corridor I would travel down. It was completely different then because it's been changed around now. But I would walk past this gallery of British natural history. It was quite a slightly old-fashioned type of gallery with odd dioramas in it. The dioramas of different stuffed animals and a few plants. You can see the bird gallery has no plants whatsoever, just birds. But the British natural history was a much more interactive thing. It was, I'd say, one morning when I was walking down this gallery, I was stopped by a family. It was an American family, and I assume the person who spoke to me was the father of the family, and he pointed to a small bird and said to me, "That's not a robin. You guys need to change your exhibition. You need to change your label." Now, my ornithology is not brilliant. It's nowhere near as bad as Terry Pratchett's here, but I'm pretty certain I knew what a robin was. But one of the things we're not told to do, one of the things we assume we're not doing is argue with the people who come and visit us. So I left that and said, well, I'm pretty sure what we've got here is a robin. This man was so insistent, it's not a robin. You need to change this stuff. So I moved on. Now, here's a robin. My view, as I'm walking back to my office, who would not know what a robin is? You see that it appears on our Christmas cards. There's a little bird there. You just wouldn't mistake this damn thing. So I looked up robins. And of course, there's this thing. It's a North American robin. Now, that interested me because obviously we were lacking some communication there. A classification that's based on common names, I guess, was the first thing you learned from that. That isn't much use really, and in terms of identification, it would be easy for me to say, yeah, we would know that's a robin. People in North America would know that's a robin. There's no need why we should know what each other's organisms are. There they are together, very different creatures. And look at those in a bit more detail, and this is what fascinated me when I started looking at these things. Again, you may know all this, and I'm just putting this out here as a beginning. What we can see is a lot more detail about this particular robin. That it's an animal, of course, in the animal kingdom. It's a chordate. It's in the class Aves. It's a bird. It's a perching bird, and so on. If we look at the North American robin, it's the same thing. It's an animal. It's a chordate. It's a bird, obviously, and it's a perching bird. But after that, it starts to diverge. So what we have here is two birds that we could say that they're actually very much the same thing, but they differ at some point. The point here that struck me was that as you move up or down the hierarchy, whichever you want, the taxonomic hierarchy, that you have different levels of commonality between different or same organisms. So what would be useful about this is that we know the upper one there is the European robin. We'd only get it in Europe. The lower one is the North American robin, which would only occur in North America. Once you know that, the identification part is simple, but it doesn't tell you what, I'm going back one slide here, it doesn't tell you what these two birds are. The one belongs to a different genus than the other. So they're two very different birds with the same name. So the identification is easy. It's got a red breast. But telling you what the thing actually is a little more complex. So one of the things that exchange made me remember, at least, is that there are three different aspects to dealing with organisms in a comparative sense. There's the identification of what you have in front of you. There's the relationships among those things that you've got in front of you. And there's how we represent those relationships. Those three things sometimes confuse. Let me present something here which I think may be something that's unusual. I tend to think that, and I was taught this, and I think this is wrong, that this is the way the process works. Here's 4 diatom species. We identify them or work out what they are or tell people, well, I've got this species, that species. There are hundreds of papers that are published on clarifying this species and that species and so on. That leads to, or can lead to, what we know about their relationships. So we can say, well, we need to know the species first, then we know their relationships. In this instance, species 3 and 4 most closely related to each other than they are to 2 and 1 and so on. Then the next step is we make a classification from that. The blocks there are, the black blocks on the branches there are we stick a few characters in there and say, well, that relationship is supported by this and that's relationship supported by that. So the process seems to be, and I'm going to emphasize this, I think this is wrong. The process seems to be that let's work out all the species first, then let's tell you how all those species are interrelated, and then let's have a classification. Why is it wrong? This is what really happens. Because of that hierarchy, animals, chordates, birds, the genus, turdus, and so on, because each of those have some identifying characteristic, really is the classification at the bottom that's first, and it leads to either or both being able to identify the series of species that you've used, or it leads to an understanding of the interrelationships of them. So the classification part is the most fundamental, it's the most important. Here, it's a departure, I think. Again, I don't know when I'm talking to the converted already, but I think it's a departure in the fact that we don't necessarily need to know all the species first. We can't possibly do that. The different characters support different parts of that tree. So the whole of the classification is important, not just the species and their relationships and so on. There's only 2 things that I've learned during my time of trying to understand how the process of classification works, and they're both horribly cumbersome characters. The first here is synapomorphy. To me, that represents any unique character that tells you that 2 things might be related. It's not hard and fast, it's a proposal, it's something that might tell you, or something that does tell you that species are definable, that they're related, and that you can characterize them. The second, synapomorphy is climbing into the diatom literature in many different guises, and I think its use is varied, not just in the diatom literature but elsewhere, but that's the first of the most important things that we do searching through specimens. The second is another horrible word, heterobathmy. It literally means, so I understand, steps on a stair. When we look at our descriptions, or when we look at our specimens, more importantly, we'll find there's a mixture of characters there. There's spines, there's raphae, there's virgae, vimines, there's all sorts of different things. What our task seems to be is not just to identify those bits and pieces, but to see where those bits and pieces fit in the hierarchy that we have. So here, in the upper diagram, there's numbers on those characters. You can see there that character 1, way down in the hierarchy, is something that's common to all the species, 1, 2, 3, and 4. Number 2, character 2, just just the 2, 3, and 4. So essentially, that's all we need to do is search through our specimens, find the characters, not necessarily just list them then and describe them, but find where in that hierarchy they fit. That's about it. This is the last I'm going to talk about anything that has a theoretical basis to it. I think that's it. We probably all know that. Let me see how that works in practice in terms of people who want to have identifications as opposed to worrying too much about how classifications are formed. So what I want to do is just talk about and perhaps avoid trying to bore you with the genus Fragilaria. Why do I want to do that? As I've said I think I wrote in the abstract that this genus has plagued me for my entire career, and hopefully I'll get to some point where I can tell you why that's so, and hopefully I can get to some point where I can tell you actually what happened along the way was everybody was correct. There never was any a problem if we focused on that notion of a taxonomic hierarchy. This bit's from Algae Base. It's nothing worse than somebody reading out a slide to you because you can read it, but forgive me for doing that every now and then. This from Algae Base, the taxonomy of Fragilaria is in disarray with some authorities splitting genus into 6 genera, including blah, 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 and others not doing so. So others lumping species signage with Fragilaria. In other words, he's saying that there's, or whoever wrote this, said that there's a mixture of different things, or else there's 2. And that to the author of this piece, I'm sorry, I don't know who it is, suggests that means everything's in disarray. I'm going to get to a point where I'm going to say that's not the case. What you see on the side here, the electron micrographs, are those taken from my first paper on this subject. They're all the usual features of Fragilaria. I'm not going to go through them because that's not the, I will later, but that's not the point of this first bit here. Investigating such things as these, I think it's always worth going back to the beginning. And in this sense, we can go back for Fragilaria, you can go back to 1819 to this book written by Lingby. This is a wonderful frontispiece he had here. I guess that must be Neptune. Again, I'm not sure how clear these pictures come across, but these are his illustrations of species in Fragilaria. There weren't that many. I can't remember how many species put in them now, 8 maybe. He has a very nice, succinct description. Still doesn't tell you what it is, and by what it is, he doesn't characterize a genus, he just describes it. Very, very different things. Here, marked out in the red box, is what eventually was thought of as the type. Here, my co-author and good friend Aki Tuji, Aki put in all the heavy lifting for this, that discovering some of those specimens, being able to look at them in the light microscope, being able to look at them in the electron microscope, so we could have some better idea of what they look like. So bringing the past to the present, if you like. Let me just look at, these are the electron micrographs we published. I've added some arrows in there just so that I can talk a little bit about the pieces. But remember, what I'm doing here, which is why this genus has plagued me, what I'm doing here is describing it. I'm not characterizing it. So the upper pictures, you see that there's one girdle band. It's an open band. It has an open part of one end. The 2 arrows point to some ornamentation on it. As you go down the picture, I'm not sure if I can point it. Does that work? Yeah, perhaps that works. This arrow here points to the spines. Look, there's some spines that overhang here on the where the pore field is. You can see the striae here and so on. Again, you can see the sternum here. The spines are the usual type. Again, here's 2 valves that are connected. Right in the middle here is where there's that central area, such as say it's slightly different. Again, here you see the rima portulae, the pore field, and so on. All of these features you will read over and over again in the description of the genus Fragilaria. What's really interesting, and Lingby did a really nice job on it, was to have another look in what he's got in his book. And here is the little bit that he wrote about put the classification at the beginning. He used this word in his section 5, homologonata, which is basically the diatoms. He had two genera, Diatom and Fragilaria. He puts the two in the same group, of course, and he relates those next door to a group of brown algae. Now, that to me is really interesting. First, what he's done is he's defined the family, or actually he's described the family. Then he's described each genus that belongs in it and tells you how they differ. You have to work out how they differ. And then he tells you how they're related. And you can see that as a kind of modern version, that all the diatoms are most closely related to themselves relative to everything else. Classification here is prior to everything else. That's what he was doing. It's an interesting diagram, this diagram, because it relates to every other kind of statement we make about organisms, whether we think we do or not. Let's borrow Terry Pratchett again. He lives in the city. I know sparrows from starlings, everything else is a duck. Well, that's the same kind of diagram. He's really just saying, and I don't know whether he means it or not, but sparrows for him, sparrows and starlings are most closely related because of some way he can identify them relative to ducks. I want to say one last thing about, one final thing about Lyngby. What's really interesting about looking at his work and devoting some time to its study is that he, just because it's 1819, he can't possibly be wrong. He's working with what he's got. And I think that's a good thing for us here. We work with what we've got. And so he has a description of the diatom group. He has a description of the subgroups. We can work through that. And he tells us how they're related based on the information that he knew. He can't possibly be wrong. That's what he knew. Here's one by Agard. I'm not going to talk too much about this one. I find this one interesting because it's some 1832, something like that. So it's, what, 12 years on from Lyngby. Agard has a different problem here. He's trying to contrast on the left here, how that diatom grows with the shape of the valve. He's trying to get that thing to correspond, and he puts the genera, he puts them in here each time to tell you what he thinks, how that's related. And he does come up with 3 groups. That's a really interesting way of doing it. Again, I don't really think we could ever judge Agard as being wrong about something. This is a really nice way of dealing with it. And he had effectively the relationships as they are down here, but also saw a different kind of relationship. So he's struggling with those two things. Agard and Lyngby were prioritizing classification, partly because that was where the place they worked in. Here's myself and Aki again. What we do is what everyone else does. We shouldn't have done it, but we did. We go on to describe what Fragilaria is like. Has typical features of Fragilaria. What does that mean? No idea what that means. And then we go on to say what we think typical features are, and we avoid defining that genus, largely because, frankly, we can't. So we've said, well, it has striae not opposite each other relative to the sternum, linking spines, open girdle bands, all those things that we still talk about, but we don't tell you what it is. Again, another guide to Fragilaria on the North American Diatom page describes it beautifully. It's exactly what you want if you're after identification, but it doesn't tell you what it is. It's not anybody's fault they don't tell you, it's that we simply don't know. Here's another one from the Diatom Flora of Britain Island. Again, it's a description. I'm not going to read out a description because all the descriptions are the same. They're very, very, very useful. I'm not suggesting for a moment they're not useful, but they describe something. Don't tell you what it is. Here's Aki and myself again. What we did later on in another paragraph is talk about Cyanidra. What distinguishes Fragilaria from Cyanidra? Everybody else has done this. I don't know why we did it, but maybe that helps. Can we, if we can't tell you what Fragilaria is, can we tell you what Cynedra is? And here we've put in brackets, if you can see somewhere, that it equals Ulnaria. I'm not going to talk about why that equals Ulnaria. This is merely a nomenclature issue, not a taxonomic issue. It's absolutely just a nomenclature issue. But we talk about later on, we talk about that it's to do with the structure of the girdle band is definitive in this case, largely because I tried to tackle this in terms of definition earlier on rather than description. Let me just try and go through Ulnaria, because that's somewhat easier, because I think I know what Ulnaria is. Cynedra, Ulnaria, freshwater Cynedras. Here, I like this quite a lot. The taxonomic history of Fragilaria leading to the establishment of Ulnaria is convoluted. I'm going to suggest it isn't. Here's a comparison between the two. I forget where I've taken this from, but the comparison again falls into that trap of trying to describe one and describe the other and say, well, how do they differ? But how they differ doesn't really matter. Doesn't tell you what they are. So what is Ulnaria? Well, this is some work I did an awful long time ago. And I suggested there were 3 characters that defined Cynedra at that time, which is effectively Ulnaria. And this was really important to me because I thought these were 3 synapomorphic unique characters. So the first was this, I put the basal siliceous layer, which was unique to these group of species. All the bands in the cingulum were closed, and there was a particular modification of the pore field, the ocellular limbus, that I felt no other genus had. So the characters defined the genus. Didn't matter what features it had elsewhere. It allowed me to see that some spined species that had been in Fragilaria before were actually part of this genus. So these were 3 defining characters, if you like. That held together reasonably well. More species were described. Here's something that Bart and Christine did from Peruvian Andes. Here's something that Eduardo did from, I can't remember where that was from now, from Bolivia. Now I've used those because they've added more characters in. I want to show you what happens when descriptions and definitions clash. Here's the list of characters. So Bart and Eduardo added a few more in. Let's look at mine. It's the typical Virgi Viminese structure. That's how I phrase it. Not very good. Closed girdle bands, cellular limbus structure, and then added in the absence of spines, solitary life form 2, Rima portuli. How can we deal with that in a rational way, if you like? Well, first, the cellular limbus structure. I think I was wrong on that. As I looked at more specimens, it was clear that didn't define this group. It defined a much, much larger group that excluded those species that had the first 2 characters. The second thing was absence of spines. Well, we can argue about that if we like, but absence of something doesn't tell you anything in terms of characterizing something. It definitely tells you something about specimens in terms of identification, but nothing about what it is in terms of what it is. A solitary life form, again, it's too vague, and it's part of the colony and how the colony form and so on. Again, it may be useful for identification, but not for classification. And 2 Rima portuli, well, that turns out not to be so. So we can eliminate of those 1, 2, 3, 4, 5, 6 characters that seemed to be defining Ulnaria. We can eliminate 4 of them. I hope you can see that what's happening here is that Ulnaria, Cynedra, was being described as opposed to defined. And my adventure with it was to try and define it as opposed to describe it. It's a tough task describing them, of course. Defining it is much more significant. Actually, later on, I had another look at that cellular limbus structure and reinstated it in 2019. I have no confidence that's going to stay, but the point there is that you guys who are going to look at it would say, no, that's not a character of that genus. Now, let me emphasize this again. We're talking at the level of genus. So the rank is important here because it's part of that hierarchy. These are generic characters, not specific characters. All right. So the point I arrived at, and the point I hope everybody else arrived at, was that I can characterize Ulnaria, freshwater cyanidras, but I couldn't Fragilaria. I just couldn't do it. And I still think it hasn't been done. Let's look at some things on relationships. We have this vast promise of molecular data, which indeed it's going to provide us. Let me just take a little trip through some of these diagrams. I've redrawn them. Again, I mean no criticism of these studies at all. I'm really interested in what they tell me. Here's the first one I want to draw to your attention. Just concentrate on this. No, I've redrawn it and renumbered the nodes because I'm interested in this one here, node 3, which tells us that in this diagram, Centronella and Ulnaria are most closely related relative to everything else. Here's a different study. It was published in the same year, different taxa, different methods, different approach. Still you get node 3, Centronella, and now includes Fragilaria, but Centronella is a kind of Fragilaria anyway. So you have Centronella, Fragilaria, and Ulnaria, most closely related. Yes, bearing in mind that, in my view at least, we can characterize Ulnaria, we can't do Fragilaria, but that node shows up again. Here's another study. This was in 2016, published in 2017. Again, a different group, different purposes, different specimens. Again, they still find node 3, Ulnaria and Fragilaria, is still there. Let me go back a bit. This is 2008. It's a paper from Linda Medlin's group. Again, node 3 is still there. Centronella, Fragilaria, it doesn't disappear, and Ulnaria, it doesn't disappear. Let's go back even further then to 1897. I'm totally forgotten who did this one in 1897. I think that's, sorry, it's gone out of my mind. But nevertheless, that's a classification. Node 3 is still in there. Here's a 1901 version. Difficult to try and work out what they mean by this set of boxes. I think this was Paragallo, but node 3 is in there. What conclusion can you make from that? The only conclusion I can come to, given that everyone looking at the relationships here used different taxa, had different methods, is that somehow this is telling us the truth, if you like. I don't mean the absolute truth. I mean, it's a repeated observation that node 3 exists. So let me just rehash what point where we've got to now, that in my view, you have to deal with it in my view. In terms of definition, we don't know what Fragilaria is. I don't know what Fragilaria is, undefinable. I do know what Ulnaria is, but somehow, whatever we include in Fragilaria is most closely related to whatever there is in Ulnaria. I said early on that my central part here was going to be that everybody was correct. Here's Horst's work in 1980, which started this ball rolling, I guess, when he put everything or a huge number of species of Cyanidra into Fragilaria and said, they're all Fragilaria. Never mind, they're all Fragilaria. This is from the abstract, the English bit, I'm afraid, because the German bit would just confuse me. The English bit, forgive me, I'm going to read this. The long suspected insufficiency of the unique decisive differential criterion, namely the potency of forming filamentous colonies, has now been proved as such beyond doubt. It's not true, is what he's saying. Frustules live singly or in band-like aggregations, linked by spines or otherwise. And the possession of a distinct labiate process combined with apicopore fields occurs in Fragilaria as well as species of Cyanidra. Yes, absolutely, he's correct. Everyone knows he's correct. What's he got there? What he's put there is put it into the same genus. Now, he'd be correct if he'd put it into a higher, a taxon of higher rank. He knows, it's obvious from the text of that 1980 paper that he knows they are different. These things are different. He knows that Cyanidra ulna, Cyanidra pulchella, tabulata, all those different kinds of Cyanidras are different from one another, but he puts them all together. But if you put them at a different rank, which is what Frank Round and I should have done earlier on, then that would be absolutely, he would be correct and everyone else would be correct. So it's a question of not paying, forgive me for this phrase, but not paying sufficient attention to that hierarchy. Let me just show you that in a couple more diagrams. Here's a nice study by Maxim on Ulnarias from Baikal. Here's his diagram from the results of his sequence analysis. The box up the top there is names of Fragilaria that we all understand. The second green box is names of Ulnaria. Again, they're each other's closest relatives. The green arrows are pointing to groups that were included in Fragilaria at some point. So they're all dotted around the place. What should happen here is this big box is what Horst was talking about. He should have called it I don't know, a family or an order or something, and then subdivided in there. So it's simply not. I don't want to use the word didn't appreciate, but I think I've never talked to Horst about this. So maybe he just wasn't tied into the idea that higher classification was worth attention. Whereas if he'd called this an order, we would have all been fine. All our job would have been to separate everything within here. I put these red arrows here because what I want to do is that the different hierarchies must have some kind of synapomorphy to support them. Here, the 3 red arrows point to where spines occur in each one in these groups. So spines is a character of this larger group. They're not a character of Fragilaria, they're not a character of Ulnaria. They occur all the way through. So that's a synapomorphy that occurs way down the tree, if you like. Again, it's find the synapomorphy. Where does it fit in that hierarchy? That's effectively what Horst was doing. He was concentrating on the node 1 of this diagram. Should have been a different rank, and what everyone else was doing was concentrating down here on the tips of node 3. So effectively, we were both right. Everyone was right if they paid attention to where that taxonomic hierarchy, where the characters fitted in with that taxonomic hierarchy. These are my last few slides here because I think this is where for me it gets really interesting. Again, I'm using this recent study of sequence data and an attempt to identify species of Fragilaria. I've redrawn this diagram. I hope I've got it right. Effectively, what these guys were trying to do is saying, well, supposing we can get some molecular data, we can show you how they're related, and then we can try and find out what defines them. What I've done instead is pull out 3 subgroups here. There's group 1 at the top, group 2, there's group 3, and there's group 4. In my view, of course, those things are separate. So Fragilaria still is not definable, even though we have these molecular data, because frankly, molecular data don't give us any concrete evidence. We get lots of figures, we get lots of numbers, we get lots of numbers, but we don't get any actual evidence that these things are related or how they're related. We get an idea of how they're related, we don't get why they are. So in my view here, that this may or may not be one group, a monophyletic group, a natural group, but there are 1, 2, 3, 4 subgroups within it. I was going to talk about this a bit more, but this is something that I've been doing. I'm still in the throes of doing it. I don't know what to do with it, frankly. What you'll see there, I've taken the picture from the plate. 10 through to 16 is a species that was named by Kutzing. This is from Kutzing's material. It was named by Kutzing as Fragilaria tenuissima. I think this is an interesting species, largely because where the arrow is here, where the arrow is there, what we have now is this very peculiar kind of girdle band to it. That, as far as I recall, seems to occur in Crotonensis and a couple of other these long skinny things. So in other words, I think what I've done is not define Fragilaria itself, but found yet another subgroup within it. So I put here that it could be my tenuissima might correspond to that node 2 or that node 3. Again, it's looking for those peculiar characters that might define the entire group, whereas all I find is ones that define subgroups. These really will be my last few slides. Again, I really have to emphasize, I mean no criticism of this study at all. It's a fantastic study. I'm looking at it through my eyes. Here they have a bunch of they've described a bunch of new species. Here's a description, fantastic description. It's very, very detailed. I'm wondering actually if anyone who's confronted by that specimen would read through all this and think, yeah, that's what I got. But it's a really detailed description. Let's just go through it a little bit. I've boiled it down to 4 characters. Rhyme portrait position, blah, blah, blah. 2 apical pore fields, spines relatively small, and no internal rim depression observed in the central area. Okay, the first one, that occurs everywhere. It's all over the place. You can't use that for identification. It certainly doesn't characterize that species. Here's the next one, 2 apical pore fields. Well, everything has 2 apical pore fields, but the 8 to 10 columns, this is a bit promising now. The spine's relatively small to round base. Everything that's in Fragilaria has that. No internal rim, everything in Fragilaria has that almost. So we're left with, like my peculiar girdle band, finishing long before the pole. Here we have another potentially unique character, 8 to 10 columns of the pore field with a maximum of 5. That's really interesting to me. That may actually define this species. Never mind whether we can describe it or not. Here's where I am, at least. And this is my close here. This is the really nice tree of relationships within maybe a monophyletic Fragilaria. The top diagram here comes from Maxim's. He has a monophyletic Fragilaria related to a monophyletic Ulnaria, maybe. Here's my effort so far. I have a monophyletic Ulnaria. I can define Ulnaria, but every other Fragilaria defies any concrete example of something that will unite it together. So to me, do we differ? Yes. Is it important? Yes. Is it an interest? Why does it matter? Because this presents a very interesting scientific problem, that if we continue to talk about Fragilaria as if it is a proper group, then we may be attributing things to it which it simply doesn't have. Now, before I do that, there's one thing I wanted to add in here. I'm talking about these as names. I'm talking about Fragilaria as a name and talking about Ulnaria as a name, and saying to you that I think where I've reached, the point I've reached is that I can define Fragilaria, Ulnaria. I can't define Fragilaria. So to me, it's impossible. I think this is an example of something that's very, very old. I remember who it was. I think it was probably Plato, who was having one of his discussions about groups. And he said that there's Greeks and there's barbarians. And Greeks, well, they speak Greek. Barbarians, they speak all sorts of different languages. And during that little discussion he has with his students about Greeks and barbarians, he says, which I think is really crucial, just because it's got a name doesn't mean it's a real thing. Barbarians are not a real thing. They're a mixture of different things. Is Fragilaria like that? At the moment, I have no idea. This is why I think it's an interesting scientific problem, because if it's not a real thing, then we're just dealing with made up stuff. Whatever you try to identify, you want to identify real biological entities. Let me just rehash these things. I think there's only two aspects to classification. One is synapomorphy, unique characters. The other is heterobathmy, fixing those characters to the right place in that hierarchy. So, for example, for me, closed girdle bands fits with Ulnaria, species of Ulnaria. Spines of any kind fits with that much larger group that both Horst and Frank Round and I missed out on describing as a, I've written it down here, I don't know, order, class, I don't know what it would be, but it's a taxon of higher rank. That would allow an understanding of where both those characters fitted. So what's that relationship between identification, relationships, and classification? I still think the arrow goes that way upwards, not downwards, that we classify these things first because that's a discovery process. We discover their relationships are discovered on the way. And then once we know what we have as biological entities, then we can describe them and have some understanding of identification. I've put 2 examples in here because of how that might work. The first is from Geraldine Reed, who is at the museum for some while. Her and I talked about her work on the on pluricin for at some length, and we discussed among ourselves what kind of classification would you have, and she settled on the one at the bottom there, and notice that she has 3 new tribes and a sub-tribe she's added in there. I couldn't tell you what characters define those. She did have them, but it's there. That's a much more useful classification than the one above, which just has 3 families and the names are just associated with that. Here's a more detailed one, which I've used again with Roicosphenia or monophyly monoraphid diatoms. Here they've put out again a really useful way of looking at it, a super order, and they've left some of these clades, some of these groups unnamed, but effectively what they're doing is going up that hierarchy. I disagree with both of those people, Geraldine and Thomas here on this work, partly because of their interpretation, but that doesn't really matter. What really matters is that the identification of that, the classification there is far more interesting and far more crucial than just having species and genera. So I go back to one of my early slides and say I think that we're doing things, generally doing things upside down. We shouldn't be looking at what are all the species, then how they're related, and then how do we define them. We should be saying, how do we get to that hierarchy? How do we understand the classification of all these characters? Then we can say, well, is this how they're related? And are these the species? I think that is my lot. Thank you if you've stayed there. Mark, I'm done.
David, thank you very much for your presentation, taking us through that journey, you know, those years of this research and describing something that's quite confusing at times and in a relatable, understandable way. So I just want to remind everyone that you can enter your questions in the chat and I'll ask David. And while we are waiting for that, I'll ask you a question, David.
Yeah.
When you showed the molecular data and those trees, you know, we still can't figure out what Fragilaria is. So in terms of those molecular studies, what is needed? Like, do we need more specimens, populations in those studies?
What do we need? That's a tricky question. I think one thing, lots of people are telling you need more specimens, more data. I don't think we do actually. I think what we need is to have some understanding of how the morphological data occurs relative to the relations expressed by those data on their own, and compare that to what we're getting from the molecules. Are they the same? Do they differ? One thing we shouldn't be doing, I'm sorry, a lot of people are doing this, one thing we should not be doing is taking the morphology and sticking it onto a molecular tree. That should stop. There's absolutely no point in doing that at all. I have no idea why people are doing it. So we need to treat morphology as if it's still independent evidence of relationships and see how it ties in with molecular data, which is a separate independent estimate of relationship. So I think it's our approach. Don't collect any more data. Let's see what we've got.
Mm-hmm. Okay, we have a question from Mark. Towards the end of your talk, you distilled shared characters down to a point of an autopomorphy. Does only possession of a unique character define a species?
it's a hopelessly unpopular view. I don't suppose there are many people who would say yes, but I would say yes. I don't see any difference. Let me be really blunt. I see no difference in rank of species, genus, family, order, so on. I see it all as diversity. I understand why we try and give special treatment to species. I'm baffled by it. So the answer to the question is yes.
Okay, I'll ask another one. In your node 3, we can
Yep.
be confident that marine cyanidra would not be in there, right?
Yes, yeah, I think so. I'd have to go and have a look. Yeah, no. Node 3 was really important to me because what it was showing was that the old cyanidra, the freshwater ones, were related to what we've commonly understood as the Fragilarias, as most closely related, and every other molecular study is showing that. So I began to understand why the early guys were confusing them as a separate piece of evidence. But the marine stuff is way down somewhere. But that's a really interesting thing that happens, is that once we get rid of everything that's not defined by those particular snapples, dump them all out, put them somewhere else, that we're left with exactly the problem that was stated way back by, I don't know who first stated it, but now we kind of have some understanding of it. I'm not convinced Fragilaria are barbarians, but it's possible.
Okay, another question here from Paula. Good food for thought. Beyond Fragilaria, are there other groups of taxa that you've been thinking about and synthesizing?
Yeah, yes, there are beyond Fragilaria. I mean, I think what's beyond Fragilaria? I mean, eternity for me, I think. Yes, I think one of the groups that's really interesting from a biogeographic point of view is all the different kinds of eunotias. There's a lot of people been having some input into that. So I think that's really developing in such a way that it needs further attention. When Geraldine Reed and I were doing the Baikal work with Amphirotia and so on, we left a stack of things undone. I'd like to get back to that because the biogeographic signal from the eunotia-like diatoms is fantastic, and that will tell us something about southern hemisphere biogeography, almost absolutely certainly. Pat's the person to ask for that. He's the guy who knows that. But that does interest me, yes.
Question from Josh. How do you deal with morphological convergence, homoplasy? Would you only be able to diagnose morphological characters through developmental studies of valves?
Homoplasy. What to say about homoplasy? A lot's been said about that in the last few decades. Let me say another unpopular thing. My impression of what's going on in the world of systematics, maybe not the world of diatoms, is that homoplasy is a little bit of a myth. I know things don't fit, but I think they don't fit because people aren't looking at those characters. So the answer to your question is, that's a great way of looking at things from a developmental viewpoint, but then you have to reconcile developmental changes with relationship changes, if you like. Let me put it another way. You'd have to reconcile development of life cycle developments with phylogenetic developments. It's ontogeny and phylogeny again. But it's absolutely worth exploring. At the moment, I have more than two abandoned papers, but two abandoned papers on trying to work out what these characters are. The central area, for example, Becky Bixby did some lovely stuff on the buttressing of Hania with some Chinese colleagues. We've just done something on life cycles there, so we can understand more about how that works, but don't know how that relates to all the kinds of central areas you find in species we call Fragilaria, species we call Cynedra. So yeah, I would love to know how the buttressing works in terms of differential silicification. So I suppose the answer to your question is yes, that'll work, and no, it probably won't.
I know that from trying to identify Fragilaria and araphid diatoms, you know, there's just few morphological characters that you can look at to separate species. Is that, you know, something that makes Fragilaria hard to classify as well because of those, there's just few characters that you can go on?
Yeah, I think that's an interesting question. There's two parts of that, I think, the interesting question. One is a general issue. Does it mean you can only classify things if you have lots of data? The answer to that must obviously be no. The early guys managed it. They were pretty accurate. That node 3 carries on right the way through. They knew what they were doing, so they didn't have masses of data. The second point is, what are the characters like in the species of Fragilaria? I admit they're not easy to work with. The Fragilaria tenuissima that I'm looking at the moment, Kutzing specimens, well, there's some unusual girdle band structure there. I guess that, and I have the time to do it. So I mean, if you want to do identification, you don't have the time to do that. I fully understand that, and it's something anyone who's going to want to do that isn't going to be interested in what I'm asking for. But if you have time to search around, how do these things differ? How do the spines differ? How do those ball fields differ? Is there something else there? I mean, the chloroplasts, what have we got there? I'm really interested in seeing how we can outline these biological entities rather than worry about identification units, if you see what I mean. So again, I'm sorry, that's not a great answer to your question. I don't think in general, not having lots of data is a problem. In fact, again, I might take the another unpopular view is that actually we can do all this with a little amount of data. We don't need these shed loads of stuff. We really don't need it because we're presenting hypotheses how things are related. So let's develop those first and let's see what we've got of those characters. But I think you're right about what you say, that the characters of Fragilaria are pushed all the way back to maybe class or order or whatever. They're not addressing the bits at the top. How do we find information for that? Interesting question. How do you do that?
I want to go back to, you mentioned biogeography earlier, and I'm sure you've looked at Fragilaria from all over the world. Do you see any insights there, different populations showing, you know, differences in morphology, certain taxa more abundant in certain environmental conditions?
My experience is limited to the collection in the museum. What I tend to see in the collection in the museum is that those slides that have been indexed for species of Fragilaria turn out not to be true. So a high percentage of the specimens in the Natural History Museum collection that are labeled as Fragilaria are incorrect. So, and very, very incorrect, even though I'm not actually quite sure what Fragilarias are, but there's, well, Bart, Luke, Hector, Carlos, they've looked at this stuff much more than I have, and I don't know, I never thought of asking the question. I will do. But that's, I mean, again, I'm answering your question at a tangent, that you look at a big collection like we have in the museum, you discover that, well, actually, many of those species or specimens that are supposed to be in some place in the world really aren't. So I suppose in a broad way, yes, you do get some insight into that. The insight is that things are not distributed everywhere. They're really not. So yes, Fragilaria does the same thing. From what we understand that to be.
Wonderful. Well, we are now at the top of the hour. Last comment here from Paula. She really likes the idea you presented of looking through a different lens or perspective at all the data out there. The patterns that emerge would fuel some good discussions. Definitely agree with that. David, I want to thank you again for contributing to our Diatom Web Academy. Wonderful presentation. Everyone, thank you for your participation, and we'll see you again next time.
It was my pleasure, my absolute pleasure.
Bye, everyone.